NER, nucleotide excision repair system. It is one of the systems for DNA repair. When there is any mutation, the DNA repair system try to repair this damage. And we have taken an example, mutation in P53. What happens when there is a mutation and how the DNA repair system repair this defect through NER, nucleotide excision repair. This protein is very important in cell division and in inducing intrinsic pathway of apoptosis. That is why if this mutation is not corrected, the cell liable to change to malignant cells. So it is very, very important to protect our body from a neoplastic process. When there is mutation, of course, due to any mutagenic conditions, chemical, physical, biological, first of all, the polymerase type 1 identifies the site of mutation, and then an enzyme which called endonuclease incises this site of mutation, how many nucleotides are mutated, excise this one, and then the exonuclease will remove this mutated segment, and then after that the polymerase type 3 reinserted the new genetic cause, which is a normal one, non-mutated one, and then this site of new genes which have been encoded in the mutated area and the mutated segments have been removed. So this new segment should be again religated to the part of the DNA chain which performed by ligase enzyme. So through this process, in a brief state, first of all, identify the site of mutation by polymerase type 1, and second, incise it by endonuclease, remove it by exonuclease, and then reimplant a new code by polymerase type 3 and religated these new codes to the surrounding genetic codes by ligase enzyme. Through this process, the whole repairing process will complete, and the new the mutation which have been took place is corrected, so the cell can perform its normal physiological activity like before.